X-linked disorders or sex-linked disorders, so sometimes there's Y-linked disorders as well, but these are genes that are found on the sex chromosomes, which are on the either X or Y chromosome. So these chromosomes determine the sex of an individual. And there's two possible combinations. If you have an X and an X, you are a female. If you have an X and a Y, you are a male. But what's interesting about these chromosomes is they are not the same size. So the X and Y chromosome are not the same, even though they match up during mitosis and meiosis. The X chromosome has a lot more genes on it than the Y chromosome, and it also has genes that are necessary for life. So you cannot live without an X chromosome. You can live without a Y chromosome. Okay? You can draw parallels to your life from that if you want. But the length of the X chromosome also determines different inheritance patterns, because let's say you have a recessive gene and it's found right here on the X chromosome. If you are a male, you don't have another X chromosome, so whatever's on that X chromosome, it's going to be expressed. If you are a female and you have this gene, you may or may not express it depending on what's on your other X chromosome. But let's say on the other X chromosome you have a different gene that is dominant to it. Well, then this gene is not going to be expressed. So this means that the likelihood of expressing genes that are on the X chromosome is different depending on whether you're male or female. So I have a example problem here. Hemophilia is a blood disorder where you can't stop bleeding. They don't have the clotting factors because of certain genes that are missing. It's found on the X chromosome. So it has different inheritance patterns based on whether you're male or female. So if a woman has hemophilia, what is the likelihood her sons will have hemophilia if her husband does not have the condition? So the first thing we'll do is talk about notation. So with X-linked disorders, we usually write the X or the Y. And then if they have the gene, you use a subscript or a smaller one, but higher. I don't know how to say it. So let's say hemophilia is the lowercase h. Okay, and she has two X chromosomes, and she also has hemophilia, so that means both of her X chromosomes must have that gene on it. All right, so what does the genotype of her husband look like? Okay, we know that he has an X and a Y, but he does not have the condition. So he must have the dominant normal condition, capital H. And then the Y doesn't have the gene on it, so we're just going to leave the Y alone. So now, what's the probability that their sons will have this condition? Go ahead and see if you can work it out. You can use a Punnett square. And then we'll talk about it together. So then going back to the question, we want to know which of her sons will have it. These on the top here are going to be daughters, right? They're going to be females, so we aren't concerned with those. These on the bottom are her sons, their sons. And how many of them, what percentage have hemophilia? All of them. So all of their sons will have hemophilia.
So with the daughters, because they have both the dominant and recessive, they won't because the dominant takes precedence, right?
Right, yeah, so the daughters will not have hemophilia. They will be carriers. Good. So let's do another pairing. Let's say one of these daughters meets up with a dude in the temple. Okay. They get married. And then what's the probability that their children, if the father has hemophilia, what's the probability that their sons will have hemophilia? Okay, so his genotype is this. Yeah, they're sons. So she doesn't have it, he does. Yeah, he does have it. Right. So again, I said sons. We're not concerned about the daughters. And one has it and one doesn't. So 50%. All right, what's the percentage that their daughters will have it?
It's 100% because one's carrying it, but one's showing it, it's 50.
Well, so they only show it if they have two recessives. All right, so yeah, 50% as well. Okay, so there you go. That's how X-linked problems, that's how to do them. They can have different combinations, but yes.
To put this into real world terms, that means that generally hemophilia will be present.
Yeah, so other sex influence traits include male pattern baldness, right? So more likely to happen in males than females. There's other traits as well. All right, epistasis. This is another kind of complicated inheritance pattern where we have two genes which interact to give a phenotype. Okay, so I'm going to give you an example one. We're going to walk through it. All right, so the gene for fur color is determined by two genes that follow a dominant and recessive pattern. Colored fur, which is a capital C, is dominant to white fur in llamas. I just picked an animal. I don't know if this is true for llamas. Okay, if a llama has a dominant gene for color, that should be a capital C here, their fur color is expressed by a second gene, okay, which is the B gene, and the big B is dominant to, which is codes for brown, is dominant to the recessive gene, little b, which is for blonde. So what are our three possible phenotypes? Brown, blonde, and white. So, what are then the possible genotypes for each? Right, so if you're white, you have a lowercase c. What about the second gene? Yeah, the second gene doesn't matter, but you could have this or that, right? But basically, if you don't have the color gene, then you can't be brown or blonde. You're just white, right? That's little c, little c. All right, what are the possible combinations for brown? Okay, so the first condition is you have to have a big C. What's the second condition? A big B. All right, so there are different ways you can get there. All right, so those are 4 different combinations which will produce brown fur. All right, and then blonde. Okay, what's the first condition you need to be blonde? You have to have the color gene, right? And then what's the second condition? Little b, right? So you can be big C, or big C, little b, little b. Oh, we missed one.
Oh, no.
You can also be that and be white. Right? All right, so let's say you take a white individual and you cross it with a brown individual like this, that's homozygous dominant. What will their progeny look like? Brown, yes, they will be that one, right? All right, let's say you took 2 brown individuals that are heterozygous for both genotypes. What will the possible combinations can they have? All right, we're going to do a Punnett square for that one. Okay, so we want to know what to expect in their progeny as far as a ratio of each. So we're going to have to do a big Punnett square. All right, what are the different combinations of genes? What are you going to put across the top and what are you going to put across the side? What's the method we use? FOIL. Good, so first, outer, inner, and last. And it's going to be the same going down the side. Okay, and then you just fill it all in. All right, and the last thing you're going to do is count up the different colors of each. All right, so how many of these are going to be brown? All right, so this is brown, 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 this is brown. I did it last class, so you know we're there. All right, so there's 9 brown. How many are blonde? this one,
Three,
and this one. How many are white?
4.
Right, these have the little c, right? Okay, so with epistasis, you don't get the 9:3 to 3:1 ratio where the two traits were independent of each other. You have one gene affecting another gene, so it's a different ratio. Okay? All right, that's all we're going to do on this.